Hi, my name is Jake, and my presentation is on CRISPR-Cas9 mediated knockout of NKX 2.5 in baboon valve interstitial cells. This project is part of a larger investigation into calcific aortic valve disease. CAVD is the deposition of calcium phosphate nodules and collagen in the aortic valve cells. This buildup results in decreased flexibility and thickening of the valve, then aortic stenosis, which is this narrowing of the valve. And as the disease progresses, the left ventricle hypertrophies to compensate for the increased difficulty of pumping blood through this narrowed valve, and eventually this causes heart failure. CAVD is a very common disease present in 20 to 30% of people over the age of 65 and nearly half of people over the age of 85. Currently, the only treatment available is surgical valve replacement or transcatheteric aortic valve replacement. Both of these procedures are invasive and risky, so our goal is to find a therapeutic target for a non-invasive treatment. One of the targets we identified is NKX 2.5. So here's why we think NKX may be a good therapeutic target. RNA sequencing data from an earlier project showed that NKX 2.5 transcription is increased in calcified valve interstitial cells. The valve interstitial cells are these yellow cells here in this cross-section of the valve. The interstitial cells are the most abundant cell in the aortic valve and play a large role in CAVD. We'll be using an in vitro knockout model to further investigate the relationship between NKX 2.5 and calcification. In CAVD, aortic valve cells undergo morphological changes that result in calcification. So as a regulatory transcription factor, NKX may have a role in this process. An interesting connection here is that a loss of NKX 2.5 is linked to bicuspid aortic valves, or BAV. This condition is when there are only 2 leaflets instead of the normal 3 on the valve. These bicuspid valves are more susceptible to CAVD than the normal tricuspid valves are. So because of this, we expect to see increased calcification when NKX is knocked out with CRISPR. CRISPR-Cas9 is a highly accurate gene editing technique that can be used to create a knockout. The knockout is produced by creating a mutation in the DNA so that the gene product is ineffective. There are 2 parts to CRISPR-Cas9. The first is this guide RNA. The spacer portion of the guide RNA is engineered to match a specific sequence in the gene of interest. Then the scaffolding part allows it to bind to the Cas9 protein. The Cas9 protein introduces a cut into the DNA, which begins the mutation process. So once they form a complex and are introduced into the cell via electroporation, the complex can parse through the DNA until it finds a section that matches its engineered sequence. Once it does so, it creates a double-stranded cut in the DNA. When a cut is made in the DNA, the cell does not repair it perfectly and may create an insertion, a deletion, or a frameshift mutation. These make the product ineffective, which creates the knockout. One challenge is that the guide RNA might not create a cut in the right place to sufficiently mutate the final product, so several may need to be tested. When working with CRISPR, the protocol needs to be optimized to the cell line. In this case, my cell line is PCAV12 baboon valve interstitial cells. These were chosen because of their similarity to human valve interstitial cells. There are 3 major steps. There's the treatment step, which involves the electroporation conditions to get the CRISPR into the cells. We need to find the correct reagent concentrations, as well as optimize the guide RNAs, as I mentioned earlier. The next step is collection. Then the development and optimization of a PCR protocol is necessary to amplify the target region to see if the knockout was successful. In the analysis step, the cells are sent for sequencing, then the sequencing data can be analyzed. Because of the significant optimization required when using CRISPR, there are 2 aims to this project. One is to develop a working protocol for the cell line, and the other is to test the effect of gene deletion on calcification. The first optimization step is electroporation. This is the technique we use to transfect the CRISPR reagents into the cell. Here we have the CRISPR reagents on the outside, and the electroporator uses electricity to make a hole in the cell membrane. When the current is stopped, the membrane reseals itself with the CRISPR inside. There are 2 variables that need to be balanced. First is voltage. It needs to be high enough to create a pore in the membrane, and then the resistance. The resistance corresponds to the amount of time that the pore is open. We will need a high enough voltage over a long enough time to introduce the CRISPR reagents, but not for too long or otherwise the cells will be damaged and die. To test survivability of the electroporation conditions, we picked 7 test conditions determined by the electric field calculation. The electrical field calculation is the voltage divided by the width of the electroporation cuvette, and the ideal mammalian electric field is between 0.25 and 3 kilovolts per centimeter. Here's a comparison of a couple of different experimental conditions, and we can visualize the survival rates. Particularly, we can see the damage and cell death occurring at the higher voltage conditions, but this improves as voltage is lowered. The electroporation results. The 1400 volts condition had good survival, but the electroporation time was very short, and ultimately we went with the lower voltage conditions here in gold. The time the voltage is applied is over 6 times longer than the 1400 volt condition, so the pores will be open for longer. They also had the best survival. With the electroporation conditions chosen, we were able to move on to experimental design. Those electroporation conditions are right here on the side. This represents a 24-well plate, and everything was done in duplicates. There are 3 conditions here. The first is vehicle only, which was just electroporation. The second was Cas9 only, and if you'll recall, Cas9 is the protein that introduces cuts in the DNA, so we wanted to make sure there were no off-target effects of the Cas9 protein making cuts where it shouldn't. And finally, we had the full CRISPR-Cas9 complex. This was done twice because I tested 2 different guide RNAs and thus need 2 separate experiments. Here are some images of the cells after the experimental treatment. This is from the 800 volt condition, and they all look pretty similar in terms of survivability and health. The other conditions looked very similar. This is just representative of all 3. After doing the experimental treatment, I extracted the DNA from the cells and preserved it to move on to the PCR development. This is necessary to amplify the region targeted by the CRISPR-Cas9. To do this, I used the NCBI primer tool to design primers and selected 3 candidate primers for each guide RNA. Since I was testing 2 guide RNAs, this came out to 6 total candidate primers. I used a gradient PCR to determine the melting temperature, and these are the 2 primer candidates that worked. So for this primer, there were 2 melting temperatures that worked, and for the second one, there was 1 here at 66.9. We expected the product to be about 800 base pairs long, and I apologize that the latter is a bit overexposed, but this band did appear right about 800 base pairs in both conditions. I also ran a concentration gradient to see if less DNA could be used per sample. I tested between 50 and 200 nanograms, but the experiment showed that the PCR needs at least 200 nanograms of DNA to work. At the time I'm recording this, I don't have the sequencing data back, but I can talk a bit about interpreting the results. This is some older data from a different NKX guide RNA in the same cell line. I'll primarily be looking here at the indel percentage and the knockout score. The indel percentage is the proportion of cells with an induced gene mutation, and the knockout score shows how good that induced mutation was at inactivating the gene product and producing a successful knockout. So if, say, we have a high indel percentage but a low knockout score, that would suggest that the CRISPR is getting in, but it's not making a cut in the right space to make a good knockout, and we would want to try a different guide RNA in that case. If we have the opposite, a low indel but a high knockout would suggest that the CRISPR is not reaching the nucleus, but it's effective when it does, and so we would want to improve the electroporation conditions in that case. Both of these scores are pretty low in this data, and I'm hoping to see improvements in both. And I will provide a live update at the end of my presentation if I have the data back. So in summary, I've determined suitable electroporation conditions and developed working PCR primers and PCR protocol. Again, I'll provide a live update on sequencing results if I have them back. If the knockout isn't sufficient, we'll go back and try different guide RNAs or different electroporation conditions depending on those indel percentage and knockout scores. Once the protocol is finalized, we'll do experiments to induce calcification in the Baboon VICs to see if NKX 2.5 deletion influences calcification levels. This full CRISPR protocol can also be used for future gene studies in this cell line. I'd like to thank Dr. Giacelli, the primary investigator and the lab that I've been doing these experiments in, my mentor Subu, as well as the group members who have provided feedback and assistance along the way. Thank you for watching.